Hi, my name is Jordan Avery. This presentation gives insight into the process of chemoreception with an emphasis on the concepts of communication and coordination within the sensory nervous system. These senses rely on specialized receptors, neural pathways, and intricate communication within the nervous system to process chemical signals, translating them into perceptible information that shapes behavior and physiological responses. First step in chemoreception involves the detection of chemicals in the environment by specialized sensory receptors located in the olfactory epithelium for smell and taste buds for taste. The binding of chemical molecules leads to the generation of electrical signals the brain uses to encode information about each molecule. In olfaction, an odorant binds to a specific receptor on an olfactory neuron. Receptors are G protein coupled receptors, and each receptor neuron expresses a single type of GPCR that is sensitive to specific odorants. Active GPCR stimulates the enzyme adenylyl cyclase to increase production of CAMP from ATP. The increase in intracellular CAMP opens CAMP-gated cation channels. This allows for an influx of sodium and calcium. This influx causes depolarization, which is conducted to the axon hillock region of the olfactory receptor. From there, the action potential travels along the axon to the olfactory bulb. In gustation, a tastant is dissolved in saliva and mucus. Bitter, sweet, and umami, these tastes stimulate type 2 taste receptor cells, which express G-coupled protein receptors called gusteucin. Activated G protein triggers phospholipase C to hydrolyze IP2 into IP3 and DAG. IP3 binds to receptors on the endoplasmic reticulum, causing the release of calcium ions. Increased calcium concentration opens ion channels, leading to cell depolarization. ATP from type 2 cells sends a signal to sensory nerve fibers. Sour and salty transduction is mediated by ion channels with stimulation through hydrogen and sodium. Sour is associated with type 3 presynaptic cells and responds to hydrogen. Hydrogen-mediated depolarization of the presynaptic cell results in serotonin release by exocytosis, which in turn initiates an action potential. Salt's cell type and transduction pathway has not been clearly defined. Current models depict sodium entering taste receptors via ion channels and initiating a cascading depolarization event. Once sensory receptors detect chemical signals, the resulting electrical impulses are conveyed to the brain through specialized neural pathways. Communication in the olfactory system begins when the action potential generated in primary olfactory receptor neurons that line the olfactory epithelium travel along their axons, which forms the olfactory nerve. These nerves project directly to the olfactory bulb located at the base of the brain, where the olfactory signals are processed by second-order neurons in the olfactory tract. These second-order neurons convey signals to higher cortical regions, such as the olfactory cortex, which identifies odors, and the limbic system, the amygdala and hippocampus, responsible for emotional response and memory formation. In the gustatory system, taste receptor cells generate action potentials that are transmitted by the facial nerve, the glossopharyngeal nerve, and the vagus nerve. These cranial nerves carry signals to the medulla, where they synapse in the nucleus of the solitary tract. From there, the information is relayed to the thalamus, and then to the gustatory cortex in the insula of the brain. The gustatory cortex is responsible for the conscious perception of taste, while the insula primarily integrates sensory modalities from both taste and smell. This slide shows where some of these signals go to higher brain orders. So you can see there's some overlap with the orbital frontal cortex, insular cortex, hypothalamus. These three structures, the insula, the limbic system, which is the hippocampus and amygdala, and the orbital frontal cortex, help in integrating sensory information from gustation and olfaction, leading to a coordination of higher brain functions that developed what we perceive as olfaction and gustation. You can see in this image the communication that occurs after transduction and how these signals are routed and what response they generate. A strong interconnection between the insula and the limbic system helps in integrating sensory information from gustation and olfaction with emotions and memory. Smells often trigger vivid memories more strongly than other senses by bypassing the thalamus and routing directly to the hippocampus and amygdala. An example is smelling a familiar childhood scent and being instantly brought back to those associated memories. The ability to remember the flavor of food and the feelings associated with it are due to the involvement of the insula. This is the primary cortical area for processing taste. The insula communicates with the amygdala and hippocampus to provide a link between emotional memories, learned associations, and sensory experiences. Damage to the insula can result in ageusia, the loss of taste. Consequences can include weight loss, malnutrition, and depression if the pleasurable experience of taste is lost. The orbital frontal cortex is another area that integrates signals received in sensory transduction. It receives input from the insula in taste transduction and the piriform cortex and thalamus in smell transduction. The orbital frontal cortex combines gustation, olfaction, and somatosensory input, including texture and temperature, to create a perception of flavor. It links different smells to specific meanings, for example, recognizing the smell of coffee specifically. It aids in assessing how pleasurable a taste or smell is, while adaptively learning and updating preferences based on experience. Damage to the orbital frontal cortex can lead to disorders such as anosmia, the loss of smell, and presbyosmia. Presbyosmia is a gradual loss of the sense of smell that occurs with aging. It can disrupt the brain's ability to integrate sensory signals that help regulate appetite. The limbic system is also impacted, which aids in the enjoyment of food, as it may not receive the same intensity of input, leading to a lack of appetite. While damage to areas of the brain can result in these disorders, other causes of these disorders include infection, various drugs, and medications. An important part of chemoreception is sensory adaptation, where the sensitivity of receptors diminishes after prolonged exposure to a stimulus. This serves an important function in maintaining the efficiency of sensory systems and preventing sensory overload. In olfaction, for example, prolonged exposure to a particular odor, such as the scent of perfume or food, often leads to a reduced sensitivity, allowing for focusing on detecting new or more important odors. This form of coordination within the nervous system prevents irrelevant stimuli from overwhelming the sensory processing centers in the brain enhancing an organism's ability to detect new and more critical environmental cues. Communication within the nervous system ensures that chemical signals are correctly detected, transmitted, and processed. The study of chemoreception enhances not only our understanding of sensory biology, but also provides insight into how disruptions in the system can impact behavior, cognition, and health.